For the past 3 years, I and other researchers have been conducting multidisciplinary analysis of dog coprolites and bones recovered from a prehistoric Illinois site known as Janey B. Goode. Dates to roughly AD 900. The site is located in the rail yards north of East St. Louis in St. Clair County and yielded over 7,000 pits and houses. Coprolites are feces that have survived the elements, insects, and time itself. They're very unusual in the Midwest. In our sample of some 150 pieces, the feces are not actually fossilized, but are instead sun-hardened and desiccated. We presume that the feces survived the elements because they were deposited during an exceptionally dry period and then redeposited into feature fills by the Indians where they then became preserved. Undigested materials found within the feces can include animal bones, chitin, claws, shells, feathers, plant fibers, hair, sinew, teeth, pollen, mucus, bacteria, blood cells, parasites, fungi, fish scales, seeds, pottery, chert, and ultimately DNA from both the dog and other plant and animal materials, including bacteria. The research objectives of this project are, one, to define the health and nutritional levels of a regionally contained dog population via parasite and bacterial analysis, as well as through blood metabolic paneling. And two, to reconstruct a gene pool through DNA analysis and understand the relationship of our pool to other domestic dog populations in North America, as well as possible affinities to coyote or wolf populations here and in Siberia. Three, to conduct a stable isotope analysis to define the range of diet, especially to confirm the presence of maize in the diet at a time when maize first appeared in this region. And four, to understand the nature of human-dog interaction at AD 900 via the eating habits of dogs and their bone pathologies. For example, how were dogs treated? Do they have fractures or butcher marks? Were they eaten? Were they formerly buried? The feces samples are first soaked in a weak detergent and distilled water. After about 2 weeks, the feces become soft and break apart. The fluid turns a reddish brown as trapped blood cells are released from the matrix. I hope you had a good lunch. This fluid is submitted for metabolic blood paneling. Macrofauna are dislodged from the matrix and are captured on a cheesecloth fine mesh screen. The material is dried and excess fluid is pipetted into 20 millimeter vials for future analysis of bacteria for bacteria and parasites. Captured macrofauna, including bone, plant, and hair fibers, sinew, etc., are dried, and they're then given to our ISAS fauna analysts for identification. Surprisingly, these dog feces have no odor. During the initial first hour of soaking, the feces emit small, trapped, odorless gas bubbles. Human coprolites, on the other hand, from the southwest, emit various gases that are anything but odorless, so this was unexpected. We did take precautions. It has not been unusual to find over 150 fragments of bone and scale from a single sample, but that in most cases represents several meals. Preliminary analysis indicate that our Janey dogs ate a variety of animals, including fish, birds, amphibians, small mammals, and rodents. They also ate plant materials. A high percentage of gar scales was found in the Janey dog feces. In one sample alone, over 65 complete gar scales were identified. Gar, although certainly edible, are extremely bony and have real dense, indigestible placoid scales. Virtually all of the scales from the site appear to be unchewed, and a few are actually burned. Associated with the scales are head bones from gar. This observation indicates that dogs were eating fish heads that had been roasted for human consumption and fed to the dogs by humans. In this sense, the dogs at Janey were not only scavengers, but were to some degree dependent on their human hosts for steady meals, especially undesired fish parts. The presence of plant fibers and dog hair has been confirmed via SEM analysis. Also, a limited DNA pilot project conducted by ISAS and the Prairie Research Institute and the Department of Anthropology and the Institute of Genomic Biology here at the university, and funded by the Purina Company, has revealed at least 12 strains of bacterial DNA in just one sample. It's not uncommon to find 1 million bacteria and multiple parasite eggs in a single feces sample. Our DNA research has identified at least one major difference in the bacterial types between modern and past dog populations. A bacteria known as bacteroidetes, very common to modern dogs, is virtually absent in our Janey population. This may be due to dietary differences. For example, modern dogs eat a very high level of carbs in their grainy foods, while our Atkin dogs 1,000 years ago were virtually carb-free.